Second genus of obligate intracellular parasites is much more difficult. It is Rickettsia, and it is genus which accounts more than count a lot of species, and actually bacteria by nature again is gram-negative, but very small in size. It is non-volatile, non-capsulated, and as well pleomorphic, means that have different shapes. So that bacteria can be transmitted only through intermediate host. Usually it is different types of arthropods such as ticks, mites, chiggers, fleas, lice, and any type of blood-sucking arthropod. So bacteria can be transmitted through generations of those arthropods and usually excreted in environments with the fecal matter of those insects. And here in dry state, it can survive for multiple months. To identify bacteria, again, gram stain is not used. Usually we're using Zdrodowski technique, GIMS or Jimenez techniques to identify bacteria. And as you can see on that right electron photo, bacteria is as well similar to chlamydia, present into different forms. It is a small spherical shape and larger rod shape form of bacteria. As well, that microorganism cannot be cultivated in culture medium without cells, and actually that bacteria is so difficult and dangerous that cultivation in routine labs is prohibited by governments of almost every country, and specific license and specific permission from governments is required for cultivation of that pathogen. That's why, again, classic microbiological methods are not applied for diagnosis of the disease. We will have some modern rapid techniques to confirm the diagnosis. And initially, Rickettsia was studied and researched by USA laboratories as a potential biological warfare. Again, as we said, life cycle of bacteria very similar to chlamydia, but a little different names. We have silent form and vegetative form. Silent form is an analog of elementary body in chlamydia, and vegetative form is an analog of reticulate body. Silent form is a small, spherical in shape, rigid to environmental conditions, form of bacteria which is highly infectious but cannot multiply. It is as well released after the cell disruption and can be transmitted from one host to another by some ways we will discuss later. So vegetative form, it is an intracellular form, rod shape, and always located within a cell in cytoplasm. It is non-infectious but highly active in metabolic activity. It can replicate, it can grow, and again, vegetative form is larger in size than silent form. If we'll talk about species of Rickettsia, again, we have a huge number of them, but usually they are classified into nowadays two groups: spotted fever group and typhoid group. Previously, we had Rickettsia tsutsugamushi, which is now reclassified into separate genus Rickettsia, but still in some books we will have three groups of diseases. Scribe typhoid group is described as Rickettsia as well. And as you can see, we have multiple species, but I gave you the geographic distribution of bacteria, and mainly disease caused by bacteria and species of bacteria causing the disease. They are limited to natural area of intermediate host of arthropod, and life cycle of arthropod or geographic area of that arthropod is limited. And diseases, they are very similar by clinical symptoms and by all mechanisms of transmission and management of the subject. They are very similar, but they're caused by different species, and they have unique names. Actually, that bacteria resembles Borrelia, as you remember, almost the same problem. A lot of different species, but all diseases are looking like, and the only difference is intermediate host and geographic location of the disease. So, of course, we will not describe every of them. We will talk only about main diseases. And again, if we will talk about possible methods of cultivation, it is chick embryo. We have a cell lines, artificially designed culture medium for that cell line, and eukaryotic cells, which are used for cultivation of bacteria. Or we have a specific line of mouse fibroblasts, which is most suitable for cultivation of Rickettsia. Of course, those methods are expensive and equipment expensive, specific conditions required, and routine labs cannot cultivate the bacteria in daily practice. So, inoculation in some susceptible animals such as guinea pig and mice is possible. And similar to Borrelia, again, cultivation in arthropods sometimes is used as a cheaper method and less problematic because actually it's much easier to feed the arthropod the tick than to feed an animal. And of course, ethic problems are less significant in community if you will use arthropods. So the same diagnostic methods, it is ELISA, immunofluorescence, and PCR test. As you can see on picture, immunofluorescence resembles other methods when bacteria is identified by glowing, intense glowing, usually in some greenish or yellow color, in the contrast with pale stained eukaryotic host cells. All diseases caused by Rickettsia have very common and almost the same pathogenesis. Bacteria, similar to chlamydia, can infect only one known type of cells. And if chlamydia can infect epithelium, Rickettsia can infect only endothelium. Of course, if disease is transmitted through blood-sucking insects, it is obvious that bacteria will be injected into the bloodstream. And of course, it's not epithelium, it is endothelium, and usually of blood vessels is the place of bacterial multiplication. And actually, any disease initially caused by vasculitis and swelling of blood vessels, which will lead to necrotizing changes and thrombosis. It will lead to ruptures of blood vessels, and actually it can lead to disseminated intravascular coagulation syndrome and occlusion of blood vessels. In clinical presentations, it will lead to different types of rashes and common immune response with blood disorders. Q fever, as we said, it is a nowadays separate genus of bacteria. It is unique in clinical presentations as well. It have another clinical appearance. We'll talk about syndromes later. But the rest, rickettsioses, have common manifestation and mainly difference in diseases only due to geographic area and arthropod which is transmitted the disease. So as you can see on pictures, we have in the left picture, it is a blood vessel which is seen as a tube, and red inclusions, it is bacterial cell of Rickettsia which is multiplying in endothelium of blood vessel. And right picture, it is a closer view of that blood vessel. You can see nucleus, dark blue nucleus of eukaryotic cell, pale blue cytoplasm, and Rickettsia cell, which are red multiplying in cytoplasm of affected cells. So most important and the most dangerous disease is epidemic typhus, which is caused by Rickettsia provachiki, which is a Czech scientist, and again, pronunciation is not English one, it's not Pravazeki, it is provachiki. Vector for the disease is human body louse, and the only source of infection, as it is again obvious, it is a human ill subject who can transmit disease by the help of blood-sucking insects. And nowadays, the disease is thought to be eradicated because actually it was most important in the time of different war conflicts, especially in World War II, when extremely low hygienic measures and no proper sanitation was possible, no medical help. A lot of people lived on the ground or in some closer to that conditions when houses were destroyed, no water supply was possible, no laundry, and actually louse, body louse was a kind of common thing. And nowadays, because of multiple measures, that disease is not present, but still it is important to know about it because bacteria, first of all, again, is kept in laboratories as potential biological warfare. Second, we will talk a little later, one more reason is present here. But disease in main course will manifest as very severe intoxication of human body with multiple organs damage, especially central neural system damage. And disease will start with severe headache. Later, it will lead to dysfunction of neural tissue, neural system with different types of consciousness affected. Severe prostration and delirium can be observed for up to weeks, and very high fever up to 39 or even higher degrees Celsius is present on those people. And of course, unique type of rashes on skin will appear. And during epidemics, especially during World War II, mortality rate from that disease was as high as 40%. And nowadays, of course, again, disease is not registered, and during proper treatment and during normal medical health, mortality rate is much lower. So it is a type of rashes which appeared during epidemic typhus. And again, as we said, disease was thought to be eradicated after World War II was over and medical help became available. Some common sanitation and common measures became available. People were checked, borderlines were eradicated, or we call it pediculosis, the state when some blood cytokinetics are present on the skin of human or in hairs of human. And actually, no cases of epidemic typhus were registered for decades. And of course, without ill person, disease cannot be transmitted and it cannot appear on our planet. And it was thought that epidemic typhus is completely eradicated from the planet. But actually, much later, few outbreaks of disease were registered in several countries, and during further investigation it became known that actually we have now it is called Rickettsia disease or recrudescent typhus. It is a state when subject infected initially by Rickettsia was not ill with acute stage or was treated incompletely because of antibiotics were not available, and bacteria was not eradicated completely. It hid in lymphoid tissue, in lymph nodes, or in lymph accumulation in different organs, and bacteria became latent. It was latent for decades, and those people they already grew. Everyone forgot that some problems were present during previous history, and later when those people were bited by borderlands, other people around them became ill with epidemic typhus. And nowadays that kind of carriage of Rickettsia by people previously ill with typhus is known as recrudescent typhus or Rickettsia disease. After that state, after the disease became known, as it's stated by multiple organization institutions, every person who participated in World War II was already checked, and no new cases of Rickettsia disease are diagnosed. Those who were identified, they were treated completely, and disease again was eradicated. So nowadays, again, epidemic typhus is stated as completely eradicated from our planet, and no ill person is diagnosed with the disease. But again, appearance of that bacteria is possible because of laboratory use of that microorganism. So very similar disease, but less severe, is known as endemic typhus, or sometimes it is called murine typhus, because that disease is caused by Rickettsia typhi and other species, and actually natural host of the disease is mice. Again, is it obvious from the name. So from rodents to human, transmission occur only through a rat flea or cat flea. So not only rats, actually domestic cats can transmit the disease to human. And in the case of endemic typhus, transmission from human to human is not acute, and actually human is a kind of dead end for Rickettsia in that case, because it's very rare, very small chance that one flea will bite human, human will remain ill and will transmit the disease to other red flea. So usually after the bite of the flea, human will be treated and the disease cannot be transmitted to other animals or other humans. But again, chance is possible. Clinical course is almost identical to epidemic typhus, but much milder duration and mortality rate is much lower. As you can see, a little different type of rashes appearing after that typhus, and in the lowest picture you can see single element by magnification by the lens, and in normal way it is looking like small spotted rash in some usually low extremity, because fleas cannot jump too high. Usually they bite in human only by lower extremities, by legs. Again, scrub typhus is caused by Rickettsia nowadays Tsutsugamushi, which previously was classified as Rickettsia Tsutsugamushi. And disease is similar by manifestation, but another vector. It is mites or chiggers, usually living in soil or different type of plants, and again accidentally transmitted to human. But by clinical presentations, they resembles epidemic typhus or endemic typhus. The only difference, significant difference, is the type of initial element. You can see that eschar is the name for the element. It resembles anthrax eschar, blackened, ulcerated area surrounded by red inflammatory zone. But those elements, of course, are much smaller and caused by the bite of the insect. So one person can have multiple elements according to number of bites, and rarely, especially if those elements are scratched by human, bacteria can invade into the bloodstream and cause generalized lymphadenopathy. In blood cell count, you will have high lymphocytosis, and in rare cases, heart and central nervous system can be involved, similar to epidemic typhus. But again, mortality rate and duration of the disease are usually much lower than in classic epidemic typhus. So another group of diseases, I will not even talk about all possible modifications. We call them just spotted fever group, because again, they all have similar manifestation. Usually ixorid ticks are the vector for that group of disease. Multiple species of bacteria, multiple different species of ticks can be involved, and actually in common disease, one or few diseases from spotted fever group distributed worldwide. According to type of the disease, course can be from very mild to even life-threatening states, but usually common symptoms include formation of primary element such as an eschar, then dissemination of bacteria by the bloodstream with severe headache and fever, and of course multiple rashes elements over the body are observed. And we have one unique disease which is known as Rickettsia pox. It is similar to natural pox, but again caused by Rickettsia species Acari and transmitted by bites of mites. Initial element will look like purple with the black crusty scab, and people notice flu-like symptoms such as common weakness, myalgia, chills, maybe fever, and of course rashes over the body. Rash is a little unique. It is popular lesions which elevated high above the skin, and again later they will be covered with the crusty scab. So Q fever is one exception, and it is one of the reasons why bacteria is reclassified, and nowadays it is Coxiella burnetii. Previously it was Rickettsia burnetii. That disease is transmitted by non-insect way. Usually it is in different types of aerosols, such as air dust, aerosol from dried feces, from urine, or even milk of different types of animal or even birds. And disease manifests with very high fever Again, common symptoms of systemic infection such as pain in joints and muscles, loss of appetite, if lungs will be involved, and usually again disease is transmitted by air, so lungs are involved. Coughing, sweating, and especially during nighttime period, bad sleep and hyperemia of face can be observed. In a few days after onset of symptoms, liver and spleen can be involved with corresponding symptoms. So during autopsy, especially during bone marrow autopsy, uh, doctors identify unique changes in structure of the bone marrow. As you can see on that left picture, it resembles donut, and actually it was known as donut granuloma. It is unique changes in the structure, which is uh, made of central lipid vacuole surrounded by histiocytes and immune cells such as eosinophils can form that unique structure looking like a ring. So again, prevention is mainly non-specific, and the most important measure here is the control of rodents, control of insects, regular checkup of especially kids for pediculosis, checkup of any person who should be hospitalized looking for medical help. Any kids collective should be checked, and any case of pediculosis should be reported, and person should be isolated and treated in properly measure. In some countries, we have vaccines against some diseases, but again, mainly number of them is very low and they're not available. So in that slide, it is a photo from National Museum of American History. It is a typhus, epidemic typhus vaccine. It was manufactured in 1952. It is a vaccine which was used after World War II, and nowadays, again, it is prohibited in many number of countries because it is a live vaccine. But still, it is kept as the reservoir for any emergency which can appear. But as well, some laboratory workers should be vaccinated, of course, if they are performing experiments with microorganisms. So if you talk about therapy, we need, of course, to use antibiotics which can penetrate in the eukaryotic cell, because some classes of antibiotics can be dissolved on the lipids and they not passing through cellular and cell membrane of eukaryotes. So here it is again almost the same tests used for identification of bacteria. It is a ELISA test for identification of antibodies, and the same can be used for multiple diseases. We have rapid tests to identify again Rickettsia spotted fever group or even scrub typhus. The principle is the same: sample window, result window, control strip, and test strip should be here. And as you can see, we can have test system to identify GM, IgG separately, or sometimes we're using combined tests when just one result will be available. And I didn't find any foreign vaccines, but in Russia, as I said, we have our own, and some vaccines which are used in Russia are prohibited in other countries because they can be dangerous. They all are alive. So in the left picture, it is a live Q fever vaccine. It contains a live microorganism which is used for skin scarification application. And in the it is a strain which is growing in chick embryo. It is vaccine strain, but still it is alive. In the right picture, it is vaccine against epidemic typhus, as well it is a live vaccine, but that one is combined. Actually, it's using two different strains of Rickettsia prowachiki. First one is a vaccine strain, which is weakened and growing in chick embryo, and it is combined with only antigen, not a live bacteria, but antigen of viral strain of Rickettsia prowachiki, and that vaccine is used for subcutaneous use. Again, only laboratory workers vaccinated in common days, and that vaccine is stored in some institutes, in the case of emergency, if some outbreak of disease will occur by different reasons.